that I'll be presenting from, and I'll pull that up shortly so that you have something then we can get started. So the name of the game today, so I know you guys have, if you have been following along, hopefully you have been following along on our journey here. We've talked about a lot of stuff. So we've talked about all sorts of concepts with evolutions. We talked about natural selection, microevolution, Hardy-Weinberg, macroevolution, speciation. We talked about the origin of life on Earth, and we talked about biochemistry. And then this is going to segue in a lot into cell structure and transport, because all of those biochemical properties are going to be really, really important today when we're talking about the structure of the cell, specifically the structure of the plasma membrane, and then how the heck we get things through that cell. Okay, so that's the name of the game for today. All right, to start, I am going to go through just the most important cell structures with you. Okay, so you don't have to know each and everything, but it is important to have a general understanding. And depending on the bio class that you're in, like for instance, in my class, I hardly touch on cell structures because I just assume students know about them from their intro to biology class. So it's really important that you remember these things, especially if you had an intro to biology class, because your teacher probably isn't going to spend too much time on them. Okay, so the first three that I have on the first page, nucleus, mitochondria, and then the rough and smooth endoplasmic reticulum. So first of all of the structures that I'm about to name are only going to be in, except for like one, but we'll get to that afterwards, are only going to be in eukaryotic cells. So our eukaryotic cell friends are the ones that have all of the membrane-bound organelles. That's a really important fact to know. Okay, so the nucleus is obviously one of the most important things that we have in our cell. It has an envelope around it. We call it the nuclear envelope, but it's really just like a phospholipid bilayer or a cellular membrane, just like what we have on the outside of our cells and the rest of the organelles that we have. And then inside we have a condensed region of DNA that we call the nucleolus. But again, all of those specific details like nuclear pore, nuclear envelope, you don't need to know about that. You just need to know that a nucleus exists. DNA is in there, it's stored in there and protected in there in eukaryotic cells, and this does not exist in prokaryotic cells. More on that in a little bit, okay? Another organelle that you need to be super familiar with, especially for next week when we go through the ever-dreaded cellular respiration, is the mitochondria, okay? So everyone knows the mitochondria is the powerhouse of the cell, right? It's not enough just to know that. We've already talked about the mitochondria when we talked about the origin of life on Earth, because we talked about endosymbiont theory, right? So the theory that the mitochondria was once a freestanding prokaryote, okay? Couple facts, and you can see this actually in this picture, is that mitochondria have their own set of DNA, which is super, super unique. It's circular, which is the same as prokaryotic DNA. They also have their own set of ribosomes, so they are good to go as a prokaryotic cell, but we use them as eukaryotes to produce tons and tons and tons of energy through cellular respiration, and we will talk about this guy so very much next week, okay? Other good things to know are about the endoplasmic reticulum, the rough and smooth endoplasmic reticulum. I have practice questions at the end. Oftentimes in the AP exam, they might ask you practice questions like, oh, this cell makes a lot of enzymes. What do you think it might have a lot of? Okay, and you would need to know that 1, an enzyme is a protein, and 2, proteins are created mainly in the rough endoplasmic reticulum. Okay, so that's just an example of when you might need to know something about these structures on a test. Okay, so the rough and the smooth endoplasmic reticulum come right off of the nucleus, and that's super important because they're going to make lots of things for us. So you'll notice that the rough endoplasmic reticulum is first. It's closest to the nucleus, okay? And it's rough because it's covered in ribosomes. So when mRNA, messenger RNA, leaves the nucleus, it will actually go right into the rough endoplasmic reticulum in order to go through the process of translation. Okay, so it's very important that those are super close because it can just shoot right over into the rough endoplasmic reticulum, which is where all of our proteins, for the most part, are made. Okay. We also have some freestanding ribosomes, and I have ribosomes on the later slide. But for the most part, our proteins as eukaryotes are made in the rough endoplasmic reticulum. The smooth endoplasmic reticulum is more where like lipids, steroids, hormones, those kinds of things are made. So not proteins, but other things, especially like in the lipid family, like steroids and hormones, like I just said. So those are going to be made there. Things that aren't requiring those ribosomes to be present are going to be made in that smooth endoplasmic reticulum. Okay. All right. Other things to know, the Golgi apparatus looks like a stack of pancakes. The Golgi apparatus comes next. So once proteins have been made in the rough endoplasmic reticulum, they'll head over to the Golgi to be processed. Okay, so this is like your processing factory. They'll cut and mold and change the proteins the way they look. Sometimes they'll add sugars onto proteins to make something that we call a glycoprotein. They'll do lots of awesome things with processing proteins in these guys. And then you'll notice that they have all of these little vesicles that are for transporting things into and out of the Golgi. So things will come and go here. It's kind of like a FedEx supercenter where like things come in, they get processed, and then they get sent out either to another destination in the cell or to the outside of the cell. Other important things, the lysosome, you can see that here in red. It's joining up with this cell and breaking things down. So lysosomes, so anytime you see the word lyse, L-Y-S, not like the lice in your hair, ew. And we'll talk about this next week with glycolysis, but anytime you see the word lyse, it means to break down. Okay, so the lysosome just breaks things down. It has enzymes, digestive enzymes in it, okay, that will help us digest food, will help us digest organelles, will help us digest dead things, things that might need to be recycled and reused. All of that takes place because the lysosome, with all of its digestive enzymes, binds with that thing and breaks it down. Okay? All right. Last two on these pages, the chloroplast. I'm hardly going to talk about it today because we'll talk about it with photosynthesis, but that's just in plants and it's the site of photosynthesis. Lots more to come on that guy. And then the plasma membrane is going to be the most important thing that we talk about today. Today is truly all about honoring this lovely plasma membrane and everything about it. And so we will learn its structure inside and out in a few slides. Okay. Last few things, vacuoles, vesicles, and ribosomes. So ribosomes, I threw it on this slide, probably should have put it earlier, but ribosomes are actually made up of their own type of RNA. So we have messenger RNA, we have transfer RNA, those are the M and the T's. But then ribosomes are actually made of something called rRNA, ribosomal RNA, and it's the site of translation for proteins. So this is where mRNA comes and binds, and then tRNA is going to join up and put the appropriate amino acid on it. And we will have, if you're very confused as to what I'm saying right now, we will have a whole lessons on DNA transcription and translation. So just stay tuned. Okay. Vacuole and vesicle, these aren't that important. Vesicles are, in my mind, more important to know about than vacuoles because they're more involved in cell signaling. But the reason I have them up here is because students get them confused because they both begin with V and they kind of sound alike. Okay, so vacuole is for storage. You can see this one is a plant vacuole, and plants will store lots and lots of water just because, you know, it doesn't rain every day. So they're going to store lots and lots of water for when they come to a time where they're going to need it. And they can also store food in those vacuoles as well. But plants are more likely to have vacuoles than animal cells, but we also do have vacuoles for storage, specifically so that we don't run out. Vesicles, on the other hand, are little transporters. They're like, if the Golgi is the FedEx center, these are the FedEx trucks, and they're dropping things off. And they're just made of plasma membrane, of cell membrane, okay? And so you can see that here, that this outer is the same as this outer, okay? So these things can actually just bind right with the cell membrane and release their contents and then just kind of get recycled in to the cell membrane, which is pretty crazy. And the same thing can happen in reverse. So this, we're releasing something, we call that exocytosis. Endocytosis, though, if we were bringing these chemicals in, This membrane could just wrap around it and form into a little vesicle and pop and deliver them where they need to go. Okay, so there's a little bit on this top. Any questions about those organelles? Okay, cool. So those are our starting points. Those are the ones that you need to know. The most important ones to know about, I would say ribosome, but we're going to talk about that at length. Plasma membrane, mitochondria, nucleus, and then the endoplasmic reticulums as well. Okay, but those are the big ones. Oh, I probably should have started with this. But now that you know about all these different organelles and these different parts of the cell, it's really important to distinguish between prokaryotic and eukaryotic cells. Because like when we get into talking about cellular respiration, when we start talking about the mitochondria, you need to know that prokaryotes do not have a mitochondria. Otherwise, nothing that I say about that will make sense. Okay, so it's really important that you have a very clear and distinct understanding of these key differences. So I have a lot written here on this poster. I have posters all over my classroom. This is just a snippet of that. But prokaryotes, so they don't have a nucleus. I know I mentioned that earlier. Their name literally means before, like kernel. Karyote is supposed to be like kernel, like think of a corn kernel. So before kernel would mean like before that protective outer layering, before the nucleus. So that's truly how prokaryotes got their name, means before nucleus, okay? Things that prokaryotes do have, though, are ribosomes, right? That's really important to remember. They don't have a rough endoplasmic reticulum, but they still have freestanding ribosomes in their cytoplasm. And they also have singular DNA. So with that singular DNA, that's going to be super important. So when we were talking about the mitochondria and how that used to be a freestanding prokaryote, a piece of evidence is that they have that single circular DNA, and this is where that's coming back into play. It's much, much simpler, and when we start jumping into biotechnology, you'll need to be relatively familiar with the plasmid and that single circular DNA, okay? Other things to note, they have no organelles whatsoever. Those two about glycolysis and transcription and translation we'll cover when we get to those topics, so don't worry too much about that. And they all have a cell wall, okay? All bacteria have a cell wall. Sometimes people forget about that. They're also usually mobile, and they're all single cellular. I realize that I didn't write that down, so you can feel free to add that to your own notes, but they're all single celled. And most of them, or a lot of them, are mobile. They can move around. So if you remember learning when you were in maybe an intro bio class about flagella or cilia, those are going to be located on prokaryotic cells, but not eukaryotic cells. Okay, so take a minute just to look through that list, and then we'll jump into how that is vastly different from our eukaryotes. Okay, so eukaryotes truly means with kernel or with nucleus. Okay, so these guys do have a nucleus. It's one of the most important distinguishing factors. It protects their DNA. This is going to lead to a lot of differences in transcription and translation when we get to that section of review. As far as DNA goes, we have multiple linear strands of DNA wrapped in chromosomes, okay? So we condense these guys. We wrap them really, really tight around proteins called histones. More on that when we talk about DNA, but very different there. We have multiple of them, and it's linear. We have lots of membrane-bound organelles. Almost everything that we just talked about is only in eukaryotic cells. We have a mitochondria, obviously, and we use this for the Krebs cycle and the electron transport chain. And not all of them have cell walls because animal cells don't have cell walls. Only plant cells have cell walls. And so plant cells are going to have that extra protection, and animal cells will not. Okay, so sometimes people will put as similarities between eukaryotic and prokaryotic cells that they both have cell walls. That's not true because animal cells do not, so it cannot be a defining factor of both categories as a whole. Okay, All right. Cool. So that's the intro to cells. That's probably the only time that I will talk about anything as basic as this with prokaryotes and eukaryotes and organelles. You just need to know it. You need to be familiar with it. I'm sure your AP teacher will expect the same of you. There are too many other things that you need to know about the cell to waste time on the basics. So grab a review book, look through the slides I just sent, replay this lecture, right? You can replay it if you're on here, and just listen to those things if you're feeling a little bit unfamiliar with this kind of stuff, because everything we talk about after this is just going to build off of those basics. Okay. All right. So let's get into the nitty gritty. So that's my basics on cell structure. Now we're going to really get into cellular transport, and this is where the plasma membrane is the be all end all. Okay. So there she is in all her glory, the plasma membrane. Okay, this is also called the phospholipid bilayer. And I can explain exactly why it would be called the phospholipid bilayer, because each of these little things, they kind of look like sperm actually, but they have like this polar head region and this like tail region. These tails hopefully look really familiar because when we talked about biochemistry, we talked about things that look like this, okay? But phospholipid is the word for this structure, okay? So this region up here that I'm highlighting with my mouse is a phosphate, okay? A phosphate is just PO4 minus. So a phosphate and 4 oxygens, okay, hanging out up here. And these guys are super hydrophilic. So hydro meaning water, philic meaning loving, right? So these guys love water. And that's great because as is shown in this diagram, there is water everywhere inside and outside of our cells. Okay, so this phosphate head region that loves water is really, really crucial here. Now in the inside, remember these are called phospholipids. So we talked about the phospho part. The inside part is the lipid part. So these guys that you see are fatty acid tails. Okay, so remember fatty acids from when we talked about biochemistry. These guys are fatty acid tails, and they're incredibly, incredibly hydrophobic. And that should not be surprising, right? Hydro meaning water, phobic meaning scared, like a phobia, right? Or water hating is what I usually call them. They're a bunch of haters, okay? But these tails in the middle that are made of fatty acids, which is a type of lipid, right? Hate water. And just remember, oil and water don't mix. So again, it should make sense that the lipids in the inside are hydrophobic because all lipids are going to be hydrophobic. Okay, so the reason it's called a phospholipid then, the phosphate head and the lipid tail should make sense. The reason this is called the phospholipid bilayer, bi, like bicycle, means 2, and we have 2 layers here. Okay, we have this top layer, and then flipped, we have this bottom layer with the phosphate heads on the outside and the fatty acid tails oriented into the inside. The reason those guys are in the inside is because they are water hating, and there's water out here, and there's water out here, okay? So they don't want to be exposed to water. They don't want to have anything to do with water. They don't want to have anything to do with anything that even likes water or is similar to water. So they're protected in the middle there, and they are going to be what makes the plasma membrane what we call selectively permeable, okay? Selectively permeable. I should have typed that on here. I'll type it right now so you have it as a vocab word. Selectively permeable. Okay. So what that means is so to be selective, like if you're a selective friend, you're choosing things, right? You're able to choose. Okay, so that's the selective part. The permeable part is the ability to either go through or not go through something. Permeate, to go through or enter something. Okay, so selectively permeable means that we are selecting who gets to come through this plasma membrane. And the fatty acid tails are really the reason why the membrane is this way. And we're going to talk in a few minutes about what things can and cannot get through the plasma membrane. All right. So here's our plasma membrane. We have these proteins that are embedded in it. So there's your phospholipid bilayer. There's that vocab word for you if you didn't have it written down, okay? But there's all of these big globby proteins, it's the best way to describe it, in the membrane that are going to help us do numerous things. And on the next slide, I'll show you all the different things that these proteins can do for us, okay? But it's important to know that not only do we have this kind of sea of phospholipids, right, which we can see for sure, but we also have these proteins that are doing other specific jobs for us within this. Because if the entire membrane was just as selective as this phospholipid bilayer, things like water would never be able to enter or exit the cell. Anything that's similar to water would not be able to enter or exit the cell, okay? So because we have these proteins that are going to allow us to transport things and do all sorts of fun stuff, we are able to, continue to live and be. But if it was just left up to the phospholipids, we would not be in good shape, okay? So more on all these proteins on the next slide. You'll see here I have an overview of 6 things that the proteins can do for us, okay? So in the upper left-hand corner, part A, they can transport things. This is what we're talking about all day today, so this is where your focus should be, okay? But there are going to be a lot of things that cannot come through those fatty acid tails, okay? So we need proteins that are going to allow those things, like water, for instance, to come in and out of the plasma membrane so that we can continue to live, right? We also need these transport proteins in order to go through something called active transport, which is a transport that involves an energy source. In this case, it's shown as ATP. Okay, more on how that's made next week. Stay tuned. Really crazy, very confusing topics. So definitely come to next week's lecture on cellular respiration. These proteins can also act as enzymes. We know by now that all enzymes are proteins, so they can start to facilitate reactions on the surface of the cell, which is really awesome. Signal transduction, we're going to talk about at length in about 2 weeks. Okay, so sometimes the signaling molecule, like this little orange thing here, signals don't actually have to enter the cell. They can just tag a protein on the surface, and that will start a pathway inside of the cell that gets that kind of like a relay race that gets that message relayed. Okay, so things that can't enter the cell normally can just signal the protein on the outside. Cell-to-cell recognition, cells talk to one another. It's a super important quality. So when we talk about the immune system, cells will recognize other cells based off of the proteins that are sticking off of them. Bacteria, when they're growing in small places, if they bump into another bacterial cell, they'll stop dividing, which is pretty crazy. But that's all done by these proteins that are on the surface. Cells can stick together this way, intracellular joining. You see that in E in the bottom middle. And then lastly, they can use these proteins to actually attach to the cellular matrix and create stronger cells, especially with plant cells. In animal cells, they have these extra matrices and cytoskeletons that they can bond to provide some structural support to the cell. Okay, so in this plasma membrane, we have all these different proteins that do all of these amazing things for us. And you have to know everything about the plasma membrane. You just need to know it. You need to love it. It's just the way it is. Okay, so phospholipids, water-hating heads, water-loving tail. Sorry, water-loving heads, water-hating tails, and then the proteins inside that can do all sorts of stuff. Okay, feeling all right so far? The extracellular matrix, Bailey, really good question, is just going to be, so some cells actually have structural support that goes outside of the plasma membrane. So it could be a channel of proteins or a channel of filaments or some sort of structural something that's just going to allow it to maintain its shape. Bone cells are going to have a lot more of an extracellular matrix, and other cells that you can think of that are more rigid are going to have other things besides just the wibbly wobbly plasma membrane holding them up and keeping them rigid. Okay, because we don't have a cell wall, we need to rely on an extracellular matrix in some cases to provide that structure. All right, so actually before we talk about concentration gradient, I'm actually going to go back 2 slides and talk a little bit more, because this is very important, about what can and cannot get through the plasma membrane, because we're going to talk about that at length and the different types of transport that different things need to use. Okay, so because these tails, and I alluded to this already, because these tails are hydrophobic, they're terrified of water, right? They don't like anything that is like water, right? So they hate water and anything like it. They're very closed-minded, these little phospholipid, or these little lipid tails, okay? They're a bunch of haters. So things that are like water, we would consider, and we talked about this a lot, so I'm not going to go into super detail because we just don't have enough time today, but things that are polar, right? Water is polar. That means it shares its electrons not equally, okay? So oxygen takes a lot of the electrons, and hydrogen are left with very few negative charges. And other things that don't share their electrons equally, or things that are ionic, like ions, anything that's charged, all of those things are similar to water. So things that are polar, things that are charged, anything like that. And those things cannot get through the membrane, or they get through the membrane so incredibly slowly that it's basically like they're not getting through the membrane on their own, okay? So I always compare, this is like a really silly example, but I always compare these hydrophobic tails to like a kraken, like an ancient squid that used to take down pirate ships and things like that. Okay, so anytime something that's similar to water, or that has a charge, or that's polar, tries to get through, it just gets repelled by the kraken tails and it has to go back out. Okay, so you can see how tight this membrane is. Nothing's wiggling through here that shouldn't be. So anytime something with a charge or something polar comes into contact with these, it gets repelled, okay? So water itself would never get through the plasma membrane if it wasn't for those transport proteins. I saw someone mentioned, Bailey mentioned, aquaporins, right? And if we didn't have aquaporins, we certainly wouldn't be alive, because water wouldn't be entering and exiting the cell, okay? So that's that situation. So all polar things, all charged things, they're just going to basically bounce right off of these hydrophobic tails and be unable to get through as much as they might like to without the help of a transport protein, okay? Now, I told you these guys were very closed-minded, these little kraken tails, these little hydrophobic tails, but they're very accepting of their own kind. So if something else is also hydrophobic, or if something is non-polar, that means it shares its electrons equally, right? Those things can freely move through the membrane. So things like oxygen and carbon dioxide, very important reactant and product of cellular respiration, which we'll talk about next week. Those kinds of things can slip through the membrane really easily, because they're also hydrophobic non-polar things, and they can just squeeze through because the hydrophobic tails have no problem with them, okay? So those things are not going to be repelled by the fatty acid tails, whereas polar things and other charged things like water would indeed be repelled by them. Okay, so those are your basics. We're gonna go through a bunch of transport stuff in just a second, and I have like a whole chart that we'll kind of go through together. But those basics, like that polar things cannot come through, non-polar things can, are going to help make the rest of our discussion a lot easier. The only other thing that is going to struggle coming through this membrane on its own are going to be anything that are relatively large. Like if we had to get a whole protein through the membrane, if we had to get a big, bulky, like a huge polysaccharide, like sugar group through, anything that's really bulky or large can't squeeze through that membrane on its own, so it will have to use a carrier protein to get through, okay? All right, so those are the basics there. We're gonna go through a whole chart that explains all of that again, so stay tuned in a few minutes. But before we do, the chart is also going to ask you guys to think about concentration gradients. Okay, so you need to know, with talking about all of this movement of substances, you need to know how substances are naturally going to move, okay, with or against a concentration gradient, because all the types of transport that we talk about in a second that involve the plasma membrane, most of them do at least, are going to talk about the concentration that things are moving between. Okay, so a gradient, first of all, is just like something that is right? Or something that has like a higher region and a lower region. So you can actually see like the grades or gradients of a hill that you might be driving down or biking up or running down or something like that. Sometimes they'll tell you some things like a 9% grade, and that's talking about a gradient. Okay, so all a gradient means is that there's going to be, you know, two regions that are different in some way. Okay, so a concentration gradient means that there are two regions that have different concentrations. Okay, so molecules always, every single time, 10 times out of 10, 100%, any way you want to say it, are going to move from areas of high concentration to areas of lower concentration. Okay, so on the left-hand side here, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18 blue molecules. And on the right, I have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12 blue molecules. Okay, so this would be the higher concentration than 18 molecules versus 12. So these blue molecules will naturally move towards an area of lower concentration. Okay, why? Really because of kinematics, and we don't have to get into like tons of details there because that's beyond the scope of AP biology, but molecules are moving, right? So they're bumping into one another and they don't want to be super close together, just like humans want their personal space, right? These guys want to freely move and spread themselves out until we reach equilibrium, which we'll talk about in detail. Okay, but that's just the natural movement of molecules. So you need to know molecules move from high concentration to low concentration naturally. When I say naturally, you can assume that means without the use of energy. Okay. On the flip side, if we were to move from an area of lower concentration to an area of a high concentration, right? Think about it as a hill. If you had to climb uphill from low to high, if you had to climb a mountain, you're going to have to exert a lot of energy, right? Same thing happens with molecules. So in their climb from low to high, they're exerting energy, and that's going to be when we talk about active transport. We can talk about that movement because it is really important. Molecules sometimes do need to move from low concentration to high concentration. Like in our brain, this is happening at all times so that we can continue to think, which is really cool and helpful. Okay. So there's lots of reasons why we would do that, but it is going to require energy. Okay. I always explain, so there's, 10 million analogies that you can use to explain this to yourself, right? But if you just think of yourself either rolling down a hill or climbing up a hill, right? Rolling down a hill requires no energy, so that would be going from a high altitude to a low altitude. Climbing up a hill requires lots of energy, so that's low to high, and that will help you remember it. And then you can always remember that molecules will naturally want to spread out because humans naturally want to spread out. If I tried to shove like 2,000 kids into my classroom, I would have to exert a lot of energy to force everyone to be together. But if I opened the door and set them free, they would willingly go, probably running, from a high concentration inside of my classroom to the low concentration outside in the hallway. Okay? Lots of different ways to remember how molecules move, but you need to know about them in order to understand the chart that I'm about to show you. Okay? All right, so before we get there, I want to mention a couple different types of movement. Okay, there are lots of vocab coming at you. There are lots of vocab. Your guess is as good as mine. This is why I don't teach English. Sorry, but there's vocab coming at you. We're going to talk about a couple different types of transport, and then we're going to organize them all into a nice table. Okay, first one, diffusion. Okay, this is like the biggest umbrella to describe how things move, and this is exactly what I just described to you. So having molecules spread out is what diffusion is, right? Now, when molecules spread out, they're moving from high concentration to low concentration, right? Because that's what allows them to reach that equilibrium. But truly all diffusion is the fancy word assigned to what I just described of molecules spacing themselves out equally, which means they're moving from a higher concentration to a lower concentration. Okay, that is diffusion. So my next three examples are all subsets of diffusion, right? So diffusion is the big umbrella that the next three types of transport and the next three vocab words you need to know fall into, okay? So up next is passive transport, okay? Passive transport is just diffusion, right? Diffusion, but this time it is across a biological membrane, right? So it's just involving a cell. So anytime you're moving molecules freely from a high concentration to a low concentration, we will call that across a plasma membrane or a biological membrane, we will call that passive transport. So it's just diffusion with one added twist in that now we are super specific because we're moving across a plasma membrane or a biological membrane, okay? The only difference between passive transport and diffusion. It's just an example that's slightly more specific, okay? Next example of diffusion is osmosis, okay? So osmosis is a special type of diffusion because it only involves water molecules. And you'll see this in the chart that we're going to fill out in a little bit. And we'll talk even more about osmosis because I have a few examples to show you, okay, with that. But water is always going to move just like every other substance on Earth from a higher concentration of itself to a lower concentration of itself, okay? But what you see happening here is that we have lots of other things, which we call solutes on the right-hand side. If we have lots of other things, that means we have less water over there, right? It's just a fact of life. You can't have as much water over there if you have lots of other stuff in there. So water will naturally move, and you can actually see it's rising up this tube. It's kind of defying gravity as it moves to push into where there's less water. Okay, so this would have been the original higher concentration of water. This would have been the original lower concentration of water. And water molecules are pushing over from higher to lower concentration towards where there's that more concentrated solute area. Okay. All right, next example of diffusion. So far we have passive transport, which is diffusion across the biological membrane, osmosis, which is the diffusion of water across a semi-permeable membrane, I should add, because that's a caveat. Oftentimes it's the plasma membrane, sometimes it's not. If you do an osmosis lab and you use dialysis tubing, that would be a semi-permeable membrane. Hopefully you do get to do that lab. It's one of my favorites. And then lastly, my last example of diffusion, the name says it for itself. It's called facilitated diffusion, okay? This is the same thing. We're moving from a higher concentration to a lower concentration. This is like a more specific example of passive transport, okay? But we are using a facilitator, right? So I am the facilitator of this session, which means I am helping you to learn cell structure and transport, okay? But the facilitator here that's helping something is either a channel protein or a carrier protein that's just allowing those molecules that can't naturally make it through the plasma membrane to get through. So everything that has a charge, everything that's polar, will use these channel proteins or carrier proteins and go through the process of facilitated diffusion in order to get through the plasma membrane. You will notice we are still moving from high concentration to low concentration here, okay? Still the same principle of molecules spreading out. We've just added a few helpers in, okay? I do want to point out the channel and carrier proteins because they're very important to know about. They both can be involved in facilitated diffusion, right? Channel proteins, like an example of a channel protein would be an aquaporin, like was mentioned earlier by Bailey, because those aquaporins, they look just like this. It's kind of like a bead in the plasma membrane, but you can see this channel, I tried to say tunnel and channel at the same time. This channel or this tunnel that is formed wraps completely through the plasma membrane, and it completely protects whatever's coming through from those fatty acid tails. Okay, so whatever this molecule is going to be directly correlated to this channel. Usually channels are specific to just one thing. So like an aquaporin is just specific to water. But it's going to let just that one thing through, and it's wide open, right? So facilitated diffusion is the only thing that it can do because molecules will only willingly move from high to low through this channel, right? No one can be forced against its will to climb up that hill from low to high because it's just an open channel, so they'll just come rushing right back through, okay? On the right-hand side, we have carrier proteins. They just change their shape. You'll see they close at the end, right, when they're being used. So carrier proteins can also be used for facilitated diffusion, but oftentimes they're actually used for active transport, because if you can imagine this in the opposite direction, forcing something from low to high, we can close the door behind them so that we don't get a flow of molecules back through, right? Because remember, molecules don't want to move that way. They're going to try to naturally slip back through from high to low. So if we're trying to force them against their concentration gradient, we need to use something like a carrier protein that can actually close behind them and force them to stay on that one side, okay? All right, so different from the rest. So we had, diffusion, specifically passive transport, osmosis, and facilitated diffusion. Different from all of those is active transport. Okay, so active, if you're active, you need lots of energy. So active transport requires the use of energy, okay? And we're gonna use this a couple times. One I mentioned previously was with large molecules, bulky molecules, things that are too big to get through the membrane on their own. And two, the time we're gonna use this the most is when we're trying to force things from low concentration to high concentration. We use energy for that. So when we talk about the nervous system, we will talk about the sodium-potassium pump. It is probably the most important protein in your body because it goes through this process of active transport. It forces both sodium and potassium against their concentration gradient, okay? And with that, it allows us to have a negative charge across our neurons, which allows us to have something called an action potential, which allows us to think, do, and be what we are, okay? So if we didn't have these guys making this concentration gradient in our brains, we would never be able to have a thought or do anything or send any signals at all, okay? So pretty crazy what these guys do for us. So active transport is very, very important, but you need to go, we're going against the concentration gradient and we're using energy to do so. So I have that all. Oh, what a concentration gradient is, Megan? So I'm going to go back to my slide there about concentration gradients, and I'll answer this quick before we move on because it's really, really important. But a gradient is just an area of change, okay? So a concentration gradient just means we have two regions of concentration. We have one area that's really concentrated. We would call that a high concentration. And we have one area that's really spread out, less molecules there, so we call that low concentration. The difference between the two areas is called the concentration gradient. So when I say going against the concentration gradient, I mean you're hiking uphill. You're going from an area where all the molecules are nicely spread out to an area where everybody's shoved in together, and nobody likes that, okay? So that's going against the gradient, like you're walking uphill, okay? When I say going with the gradient, oh, this is cool. I should use this pointer more often. When I say you're going with the gradient, you're going downhill. You're rolling like this naturally would downhill from an area of high concentration to an area of spread out low concentration where things can be more spread out and have more space. So with the gradient is going down. Against the gradient is going up the hill from a low concentration to a high concentration. Okay. All right. So this table is super, super important. Know it, love it. Put it under your pillow at night. I don't know. But it's going to explain everything that I just said. So we can take a look at it each column. And this will help you organize your thoughts and help you answer any questions with regards to cellular transport. Okay. So it's really important to know which type of transport require energy and which type do not. Okay. The only one that requires energy is active transport. Osmosis, facilitated diffusion, diffusion and passive transport do not. Okay? Sometimes students think just because facilitated diffusion requires an extra protein, and it also requires energy, and it does not. Okay? So osmosis, right, is like a glorified example of facilitated diffusion sometimes if it's across the plasma membrane. If we had to expend energy to get water through the membrane, we would be dead in like 2 seconds, okay? Because water just needs to naturally move across the membrane as it does. And so we can't expend energy on it. We just need to give it a little protein helper, a little pore or a little channel to get through so that it doesn't have to interact with those hydrophobic tails. Okay, so active transport is the only one that's going to require energy. Molecules transported. I have vocalized all of these by now, but here they all are in one place. So diffusion and passive transport is the small nonpolar molecules, right? Because nonpolar things are A-OK by those hydrophobic Kraken tails, okay? So they can get through no problem without any sort of extra protein. They just slip right through. Examples, carbon dioxide and oxygen. Facilitated diffusion, again, we're still moving from high to low. I'm going to add that column in a second. Okay, but now we're talking about the polar molecules, right? The things that can't get past those hydrophobic tails, okay? Those things are going to need to rely on a transport protein, and so they go through the process of facilitated diffusion. Active transport can be for any type of molecule if you're trying to move it from a low concentration to a high concentration, but it can also be used for just bulky things that are large proteins, stuff like that, okay? Osmosis at the end is just for water, so don't mix it up with anything else. It's really just an example of facilitated diffusion, but it's extra particular, and why not throw an extra vocab word at you? Okay. Last column to add here. This should be super, super straightforward because anything that doesn't require energy is moving from a high to a low concentration gradient. So from an area where everything's really crowded together to an area where everything's nice and spread out. Okay, so that is true for diffusion, passive transport, facilitated diffusion, and osmosis. The only thing it's not true for is active transport, which is pushing things up against its concentration gradient. Super important to know about that because next week when we talk about cellular respiration, we'll talk about how the electron transport chain uses active transport and where it gets that energy from. It's pretty crazy. Okay, questions about this table? Okay, cool. So water, just as a caveat, because sometimes the students get confused about this, water, just like everything else, is going to move from a high concentration to a low concentration of itself. Okay, it just so happens that that also means it's moving towards where there is more solute. Okay, or when I say solute, which I will talk about kind of a lot because we're about to talk about iso, and hypertonic solutions. Okay, so when I talk about solute, what I'm talking about is other things that are dissolved in water. Okay, so you can see these little purple molecules are all dissolved in water. They actually have water molecules wrapped around them. Okay, and so there's less free water there. There's less free water available because it's being bound up by the solute, and there's just less room for water molecules if there's other stuff in it. Okay, so over here we have pure water. It's just 24 free water molecules. Over here we only have 4, right? Because everything else is bound to a solute. It's not free floating. So water is naturally going to move from this area where there's 24 water molecules into this area where there's only 4 water molecules, from high concentration to low concentration. And that will always correlate with where there is more solute, okay? Because where there is more solute, there is less free water, okay? That's something that you might need to tell yourself like 10 or 15 times in order to fully understand. Okay, but where there's more solute, there's less free water. And so free water will move into that area. Okay, now this movement of water, depending on if it's moving into or out of cells, we got more vocab words, okay? Because why not just add even more? And we're not even going to have time for the practice questions. I'm just going to zoom through hypo, iso, hypertonic, and I'll give you my examples of how to remember those things, but you need to get that vocab down, okay? So this is all talking about the movement of water now. All right. So these are animal cells, these are red blood cells, okay? And there are 3 types of environments that we can say that cell is in, okay? The cell itself has not changed in each of these, but the environment that the cell has been put in has changed. Okay, so the environment that it is, we can call it hypertonic, isotonic, or hypotonic, okay? So hypertonic means that in this environment, water is moving out of the cell, and we can see that down at the bottom. Okay, so these cells are all shriveled up because water is moving out of them. Okay, now you have to remember that water always moves from an area of high concentration of itself to an area of low concentration. Okay, So what must be happening here if this is hypertonic and water is leaving is that there is less free water outside of this cell than there is inside of this cell, okay? If there's less free water outside, we can also assume that there's more solute outside, because if you remember from the previous slide, where there's less water, there's more solute, okay? So a situation like this, maybe these cells are in really salty water. You can actually dehydrate yourself. You can actually die from drinking salt water, right? Because if you do, your cells are now surrounded by lots and lots of salt, which means they're surrounded by less free water. So all the water inside of the cell is going to be like, hey, there's less water outside of me. I'm going to run outside to where there's less water, and all of a sudden your cells are all shriveled up and you can die. Okay, so that's pretty crazy. So that's hypertonic. There's more solute and less water. Same thing, more solute equals less water outside of the cell than inside of the cell, okay? So water leaves. The way that I remember hypertonic water leaves is when you're really hyper, you go run outside, okay? When you're really hyper, you go run outside. So in a hypertonic environment, water runs outside. So if you can remember that, and you remember that things move from high concentration to low concentration, then you'll always remember what that means for our hypertonic cells. In the middle, we have isotonic, okay? Think isosceles triangle, 3 equal sides. Isotonic, iso means equal, okay? So this means the amount of water inside and outside of the cell is equal. So if there was salt inside of the cell, there's an equal amount of salt outside of the cell. And so water is just going to freely move, as all molecules do, a little bit over here, a little bit over there. And so we see no movement of water, but we call it no net movement, because all the water that leaves comes back in. And we just have happy cells. So for us, for instance, we have 0.9% salt inside of our red blood cells. So when you get an IV, if you've ever had an IV before at the hospital or at the doctor's office to pump up your fluids, they're not pumping pure water in there. Otherwise, our cells would explode. Stay tuned for that in a second, okay? They're actually giving you a solution that's 0.9% salt so that your cells maintain a isotonic environment. We don't have shrinking cells or exploding cells because if you're getting an IV, you certainly don't need any other problems, okay? The last one then is hypotonic, okay? Hypotonic, and here this is the opposite of hypertonic. This time water is moving into the cell, okay? And in this case, we can actually have cells that burst, that explode. So if water is moving inside, it means that there's less water inside, right? Because water is going to move from high to low concentration. So reasons why there might be less water inside than outside, right? If you put pure water on a cell, right? There's not just pure water in cells. Like I said, our cells are 0.9% salt. So if we put 0.9% salt cells in pure water, this is 100% water. This is like 99% water. The water is going to move from a higher concentration into the lower concentration. So if you put anything into pure water, it will be hypotonic and it will get bigger and it could potentially explode. Okay, so the way that I remember that, hypo-hippo. So hippos are lazy, they're big, right? They're big, so they swell up like the cell swells up, but hippos are also lazy, so they come inside to go take a nap. So when you're hyper, you go run outside. If you're a hypo-hippo, you come inside and the cell will swell up because more water has entered it. Okay, so those are just a couple fun ways to remember these concepts. Okay, but it's all about the concentration gradient. So I always have my students draw pictures of this out for themselves so that they don't forget it. Okay, all right. I did want to show you, and then you're going to just have to look through the practice questions on your own, and I can answer questions next week if you want. But I did want to show you what this looks like in a plant cell, because it's a little bit different. Plant cells actually kind of like being in more of a hypotonic environment where water is entering. One, they have this big old vacuole, which we talked about in the beginning. And two, they have this cell wall. So they're actually able to not explode if they're a little bit overfilled with water. Okay. So if you've been to the grocery store and you're in the produce section and you get misted by the little sprinklers that they have over there, they're really spraying those plants because then there's a hypotonic environment, more water outside than inside of the cell. So water will enter and those cells will get bigger and your vegetables will look crispy and fresh. Okay, so now you know the reason behind that. In the middle here, plants can still be fine in an isotonic environment, but they really, really struggle in hypertonic environments. So if you water a plant with salt water, they're going to be in big trouble. They call it plasmolyzed because water is just being drawn out. It gets pulled away from the cell wall, and we are in rough shape. Okay? Any questions about those three? All really, really important environments to know about. And again, if you do an osmosis lab, you will see all of those in action. But yeah, hopefully those little acronyms will help you remember it for the rest of eternity. Okay, so definitely feel free to like replay some of this stuff if you're confused or just need to hear me say it again. That's what the replays are for. And encourage your friends to sign up as well because the more people we have in here, the more or the stronger discussions that we can have, and the better off we'll be. Okay, cool. So that's that. That's all I got for tonight. Next week, you have to be here. We're talking cellular respiration, so tell your friends. There's not a single student that I know that doesn't need extra help with cellular respiration. Okay, so even if you haven't learned it yet, you got to tune in. It's super, super important. It shows up on the AP exam a lot. So be there, be square, tell your friends, get them signed up. It's $15 for the whole year. You can't beat that. That's like going to, I don't know, Panera or something like that. Okay, so definitely have them signed up. And I will see you all, hopefully actually see you with my video camera next week. Okay, see y'all later. Have a good night.